All right. So welcome everybody to our May Microbiology Journal Club. And today we're going to be talking about the design and synthesis of a minimal bacterial genome. There's our paper. You can see already I have covered it in notes. So we have a lot to talk about. We also posted the link to the previous paper, which is Creation of a Bacterial Cell Controlled by a Chemically Synthesized Genome. So this minimal bacterial genome paper, which is kind of the focus of what we're talking about today, this came out in March of this year, builds very heavily off of this paper. So we have this kind of in reserve to point back to if we want to kind of think about the ways that they've constructed this minimal cellular genome. So before we launch into this, I'll just have us do introductions. At this point, people have probably introduced themselves a couple times, but we'll do it again. My name is Laura Williams. I'm an assistant professor at Providence College in Providence, Rhode Island, which is a primarily undergraduate institution. We're done with the academic year, hooray! But on Monday, all of our summer research students arrived. So what's that?
You made it.
I made it, I know. This is my first full year. I made it. I lived. But on Monday, our summer research students arrive, and I have 5 of them over the summer, so it kind of felt like this much of a break. But we're going to launch into our research season in the summer, which is when I think a lot of people, regardless of what kind of institution you're at, I think that's when a lot of people get a lot of work done. So we're very excited about that. So then I will have Nicole go. Nicole, do you want to introduce yourself?
Hi there, I'm Nicole Sictio, and I'm a postdoc at the University of Northern British Columbia in Prince George, British Columbia, which is primarily undergraduate, but does have a grad program in the biochemical sciences and in natural resources environmental studies. So I look at soils from an Illumina sequencing perspective to see what happens with fungal and bacterial communities when you rearrange the upper 20 centimeters. And that's what I get paid for.
Excellent. I think that is that's a good like short pitch for what you're up to. So let's have Dave go next, and then I saw Karen pop up, so we'll have her go last. So Dave, do you want to introduce yourself?
David Baltras, University of Arizona. We just finished the academic semester too, and so I'm calm now. It's good. I've got a nice 2-month break before I have another kiddo. So, yeah, I'm interested in microbes and adaptation and genomics and pretty much everything bacteria do. So, I get way too distracted with things, but it's fun.
I was just about to say, at this point, you just have no idea what you're interested in, are you? You're just like, everything. Let's just do everything. I don't care.
Exactly. That's my strategy.
Did you submit your tenure packet?
In a month, I think. It goes in a month.
Wow. Okay. So, well, good luck with that. So, tenure packet, kid. You know, it's an easy summer. It's a relaxed. No problem.
You got another grant. It's fine. No, so that I look at it as this. If I was gonna have a hard time with tenure, they would have probably let me know a little bit by now. So. They're letting me progress through the process. So.
I would certainly hope they would have told you by now. Yeah, that's how it should go. So hopefully that's just kind of not something you have to worry about.
Well, as long as I don't piss off anybody in the outside world and they write bad letters, I think we'll be okay. But.
So don't say anything too inflammatory in this journal club. Great. Okay, so and Karen, I just saw you pop up there. Do you want to introduce yourself?
Sure. My name is Karen. I'm a PhD student at Simon Fraser University in Canada. Can everyone hear me?
Yes.
Okay, that's good. And I work on bacterial virulence and a pathogen called Francisella tularensis and virulence factors related to Francisella. My interest in the paper is usually because, like Dave was saying, I'm very interested in microbes and all the things that go on with them. And I still haven't figured out exactly where I want to direct my future research, so I totally understand how that feels. And that's my interest in the paper as well.
And where are you with your, this is probably not a great question to ask you right now, but where are you with your dissertation? You're close, right? You're going to defend relatively soon.
Yes. I don't have a date yet. I'm just working on finishing this last figure. I have this last Western blot figure that's driving me absolutely crazy. But as soon as that is done, I can finish my last chapter, and then I should be done somewhere in anywhere between the middle of August to September.
Excellent. So, Karen, on the market looking for stuff, if anybody's watching. All right, well, very good. So, we're going to launch into this paper. As a point of semantics, I guess it's semantics, I did want to point out, like, this is the 2010 paper that they built this, this most recent paper off of, where they basically said, can we chemically synthesize a genome of a mycoplasma, put it into another mycoplasma cell, and have it actually work? And they did that. This word, it's just kind of a funny little note here. When my very first research internship that I did, I think it was my second in college, like in the summer in between college, I worked for this guy named Don LeBlanc, who's an awesome guy who worked at Eli Lilly. And I was writing something up for him, and I used this word creation, and he was like, no, like, never say creation. You're never allowed to say creation. You're not God. I was like, so I saw that go in there, and I was like, oh no, that's a Don LeBlanc red flag. But there they are.
My guess is that the title was something like, you know, we created life, and then somebody called them on it, and they had to change it to make it more subtle.
That's why this says design and synthesis, and not creation. So that was a total aside, but it made me think. It made me think back to some of my first scientific experiences and research, and that kind of made me chuckle.
You know, photoshopping though, really, when you think about it.
Do what?
It's almost like Photoshop with the genome. That's kind of how I see the paper. There's a lot of cutting and pasting and taking out a helix because they want to see if that's going to work, you know?
Yeah.
I mean, airbrushing a model, but not really. I mean, and for the sort of the same reasons, the contours in terms of gene content, which is a very bad metaphor, but
yeah, no, I think that's true. I think it's true. So you're not I don't know, you're not kind of building something from whole cloth. So you're not basically saying, we're going to create something, I just used it, so I got the Don LeBlanc red flag. We're not going to construct something from scratch that's not ever been seen before. But I think with the 2010 paper, they were just trying to figure out, can you synthesize DNA, put it into a host cell, and will it do everything that you would expect a naturally occurring chromosome that was a product of replication to do?
Absolutely.
But I think the key thing, and maybe because Dave actually suggested that we have this paper linked as well, and I think it's because the most recent paper, because it builds off of this synthetic genome, there were things that they did to tinker with this synthetic genome that I think is important to keep in mind. I don't think we need to go into like excruciating detail about them, but there were some places where they added sequences. There were some places where they do note that there were a few kind of spontaneous changes that happened as a result of this like complicated process that they went through in order to assemble a synthetic genome. So that's what they're.
My favorite story from that first paper. This is I love this paper as an example of how science actually works. And I use it in my class, but I build up from the beginning talking about restriction enzymes, and all the students are like, why do we have to learn about restriction enzymes? And then at the last class, I bring in this paper and be like, look, they just wasted 2 years because they forgot about restriction enzymes and not being able to actually synthesize things. So it's I just love the fact that they put so much effort and money into it, and then it's like methylation that causes them problems in the end.
Yeah, absolutely. So what you're mentioning, if people wanted to find that, is there's a part where, and now I'm not going to be able, I spotted that too, and now I'm not going to be able to find it quickly on the paper itself. But what they basically said was they started out with, what they did was they synthesized a genome that's typically found in one species of Mycoplasma, and they were going to move it into a host cell from another species of Mycoplasma. But in the meantime, they actually did a lot of the assembly in yeast. But yeast don't have the same methylases to modify the DNA as these two mycoplasmas did. So when they took it out of the yeast and stuffed it into the Mycoplasma host cell, the new host cell basically said, and like chopped it into a bunch of little pieces. Just like Dave said, I mean, I don't know, how did they spend 2 years? They spent a long time basically going, why is this not working? And yeah, they forgot
It was a
about
long time.
it was a long time.
And then it was funny though, because then Ham Smith is like Ham Smith is on the paper and is heavily involved in it, right? And then that's you wonder like what would have happened if he was, because I'm sure like he called out something like that, like, oh, we should have thought about this or something. Because Ham is stupidly brilliant, like, and knows all that stuff. So
yeah, the other thing that I liked too was the bit where it says, this is just great, that they were basically like, initially an error-containing clone was used to produce a synthetic genome that did not transplant. This was expected because the error was a single base pair deletion that creates a frame shift in DNA A. So there was one change that just like completely ruined the I think one of their clones, and it sounds like it took them a little bit of time to figure that one out too. But the upshot of this paper is that what they ended up with was about a 1 megabase pair Mycoplasma mycoides genome, which they're calling JCVI SYN1, that is replicating and continuing to propagate happily in Mycoplasma. Is it capriculum or capricolum? Does anybody know how you're supposed to say that? My Latin's terrible.
isn't it a Capricorn? Because that's like goat or something, right? It's yeah, I think it's Capricolum, but
okay, Capricorn. As long as I make it through this without calling it like Capri Sun or something like that, I'll be.
No, it's like the stars thing, the constellation.
Capricorn?
Yeah.
Capricorn? Capricolum? Okay. That sounds good. We'll go with that. All right.
You can tell I'm tired, by the way. So sorry about that. Like that constellation thing.
That star thing, you know. It's all good. This is not an astronomy journal club, so you're okay. All right, so that's this, and we might refer back to this if we need to kind of get some orientation. But so what they released in March was this paper, which they basically used the JCVI SYN1 genome as a template to try to ask the question of what is, in this context, the minimal genome required for Mycoplasma cellular life. So caveat upon caveat upon caveat, like this is heavily context dependent, which they do discuss in their discussion. If you're going to start talking about the minimal genome for cellular life, you have to be very specific about what do you mean in terms of the circumstances in which that life is occurring. So if you're talking about a flask in a laboratory where you can supply lots of nutrients, lots of small molecules, and so forth, that's one thing. If you're talking about actually occurring in nature where there is stochastic effects and changes in the environment, that's something else. And they do mention that, so they're aware of that, and they make that relatively clear. Before we launch into this, I just thought I would ask if anybody's got any overall impressions or thoughts that you wanted to kind of get out before we start to kind of dig into the details in terms of thinking about the way they framed this and the kind of the goal that they're after in terms of minimal genomes. Any thoughts on that?
I would love to be able to do science like this. If I just could get like people to give me money to do it.
How did they? I mean, is this? I didn't actually look. Is this federally funded? Did they do it themselves? Is it DARPA?
Yeah, so it's part of Craig's empire. So it's just private investors, and there might. I mean, they get federal grants there too, but it's there's a lot of private investors, and there's like, he got like a $600 million grant from Exxon or something to basically make biofuel stuff with this. So it all goes into the same pool. So.
Yeah.
I wish I could do that. Like, there's only one place in the world that can actually do this. So.
Well, and it honestly, the way they frame this, and I mean, they're writing this from their perspective, so I could assume that this is not a story that they're spinning now. I mean, they frame this as if to say, like, this has been one of their overarching goals for their major research programs for a long time now, is to try to get at this idea of what's a minimal bacterial genome. How do you break something down to its essential components, I think, like you just pointed out, with the eventual intention of maybe being able to add pieces back in for different industrial functions. You know, can you make the perfect oil-degrading bacterium? Can you make the perfect, you know, what is it like? Isn't there a dye? Maybe it's indigo that's made by bacteria now. So can you, in fact, engineer, if you break something down to its essential components, can you then engineer it back up to be the perfect little machine for making things for industry? Which
Well,
that comes later.
This might not be the time, but I mean, just the idea, jumping forward, probably because I'm trained as a biochemist, is the idea of like that thing that you said, the context, and the interesting thing about that very last section of the paper. I mean, it's a baby with no immune system. That is what they made. You have to feed it cake, and you can't let anything else near it. I mean, so it's viable. I mean, you can calculate a doubling time, you can grow it in culture, you have it. You can get colony morphology, but the fact that things like large repertoires of transporters or, well, membrane proteins were gone. So the idea is about systematically putting obstacles in its way. What is it going to do if you have largely polymeric substrates in the environment? What are you going to do if you co-culture it with something else? What is its defense, if it has a defense response, look like, you know? So the ideas of the bioprospecting, can we use this to engineer synthetic things, but also looking at what are the minimal gene sets needed to be viable when your environment cannot give you small, processable, easy to find things all the time, I think is also one of the interesting implications here.
Yeah, definitely. I think that's a really good point. So it kind of, when you were talking about that, oops. No, there we go. When you were talking about that just now, it made me think of the boy in the bubble.
Absolutely.
So it's like, and I read a book by Rob Dunn where he kind of mentions that in the context of understanding how your microbes are important and so forth. But it's the, you know, he had no immune system, and so I think he was raised until maybe the age of 12 or something.
Yeah.
Just in a bubble. You can't interact with anybody. You can, you know, you have to be isolated. You have to be given everything very. That's kind of what they've made. They've made the bacteria in the bubble.
Yeah, but for like, so for making yogurt or for making vaccines and stuff, where we're growing everything in very structured, sterile environments anyway, right? So if you're going to make a big fermenter, you know, you don't necessarily, you only need to do what you need to do.
Yes, yes, and I mean that's a utilitarian context. I think I'm thinking about the wild, you know, the dirt, you know, the heterogeneous environment. So like this sort of, I guess, sets the stage for, are we accessorizing or are we changing protein function? I mean, this is a question that you can't really address through this study, but it's this idea of, does the repertoire need more genes, or does it need genes that have other specializations? You know.
Well, this gets to the whole idea of, like Laura was talking about, one of the biggest, I think, the widespread critiques with this whole program is that it's so context dependent, where you can make a minimal genome, but it's really just a minimal genome for the context you want to grow it in. So if you look at John McCutcheon's stuff, there's much smaller genomes, but they can only proliferate inside of bacteriocytes and insects. Or, you know, if you put it, the minimal genome is defined by the environment it has to survive in, not necessarily by you know, core functions. So.
Yes. So we're actually talking about the resource context that permits viability, which I guess is the question of the hierarchy of where you list your genome versus your environmental list of ingredients that would support life.
Yeah, and I think in addition to that, I think that's one of their goals, is to kind of understand that. But then another goal that they mention is to try to, and this front page is basically just a summary of what comes next with the paper. So some of it reiterates their points, but they do mention here kind of our goal is a cell so simple that we can determine the molecular and biological function of every gene. So this gets back to, I have at times had my little mini rants about how can we still have proteins that we don't know what they do. And that's one of their goals here, is to try to figure out assign a function to some of these genes. However, I put a little note next to that. As we're thinking about things like resources and context dependency, another thing we have to think about too is if you divorce genes from their overall genomic context, you might lose some important information about what their biological and molecular functions are. So you're going to be able to potentially suss out a larger function or a broader kind of class of function for some genes, but if you pull it out of the genomic context, there's a chance you might miss some important interactions that that gene product has with things that you deemed to be non-essential and then removed. So I think that's something else to think about. It's not a fatal flaw in this, but it's certainly in the continuity of what we're talking about with context dependency. Just assigning gene function also has some context dependency too. So I think that's an important thing to keep in mind.
But do you think they threw that in afterwards, like once they realized that a lot of the genes that were needed in the genome weren't identified, that they're like, well, now we can figure out what they're good for? So I don't know if that was their original goal, was to define each gene's necessariness for life. But
yeah, that may be true. That may be true. You're right, because they did find that, and then that could have put that in there. Okay, so Joel, do you want to, since you popped in, do you want to just really briefly introduce yourself?
Oh yeah, I'm Joel Graff from Montana Tech, and my usual intro is that this isn't really my sort of research, so I'm going to ask lots of stupid questions along the way. I'm more into medical infectious disease host pathogen interactions is more my field of study. So I'm here mostly to learn and ask silly questions.
I think that's an important role. Okay, so all right, so we talked a little bit about the overall approach that they're using here. So, you know, they spend some time in the introduction kind of setting up this idea of a minimal genome, setting up the idea of what they're going to attempt to do, which is define genes that they're going to deem, in this context, non-essential, and they're going to set about trying to eliminate them. They come up with a couple of terms that we'll get into, this idea of non-essential, essential, and then they introduce this idea of quasi-essential, which, okay, so we'll talk about that. But I think one of the things that we might want to do is discuss a little bit about how they went about this, because they started with their synthetic genome, and then they went through they describe in the paper what sounds like, you know, somebody already mentioned that this is probably going to give some poor graduate student ulcers, so probably quite a few people, you know, probably they had a lot of stress with this, because it sounds like they went through a lot to try to get to this point. Basically, they came up with a few rules for how they decided they were going to delete genes, or how they went about deleting genes, and I think it might be useful to just kind of set those up so that we know their general strategy for how they did this. So on page, oh boy, so now they've got, I think I've got it listed as page AAD6253-2, which sounds like an antibiotic resistance gene, but apparently is a page number. They talk about how they developed a set of rules for deleting genes. And they go through this in some detail, so I think I'm just going to mention this briefly, because I think it is important to think about how they did this. So they said if they deemed a gene to be non-essential, they deleted the entire coding region, including the start and the stop codons. If there was an exception, they kind of go into it then. So they said, okay, well, when a cluster of more than one consecutive gene was deleted, the intergenic regions within the cluster were deleted also. Intergenic regions flanking a deleted gene or a consecutive cluster of deleted genes were retained. So they're kind of trying to explain how they went about keeping or deleting intergenic regions. Then they said if part of a gene to be deleted overlapped a retaining gene, that part of the gene was retained. If part of a gene to be deleted contained a ribosome binding site or promoter for a retained gene, that part of the gene was retained. When two genes were divergently transcribed, we assumed the intergenic regions separating them contained promoters for transcription in both directions. And when a deletion resulted in converging transcripts, a bidirectional terminator was inserted if one was not already present. So they tried to think a little bit about gene expression and regulation. But I do think one of the things to consider is if they're taking out intergenic regions, if they're taking out regions that they may not actually know what kind of role they serve for regulation, there could be some downstream impact that they're not going to be able to fully characterize. So they had to come up with a set of rules, but I felt like it was important to keep that in mind, this idea of what are they doing with these intergenic regions? Should we be thinking about what might happen because they're removing some of them? I don't know if anybody had any thoughts on that one. Have it like, nope, intergenic regions, yep. Okay, so then they went through this whole process where they started to, I like their thing that they said, that the first section where they're like, preliminary knowledge-based hypothetical minimal genome design does not yield a viable cell, which is probably one sentence and a whole section that was years and years and years of somebody's work, where they basically said, you know, we had a little bit of information from the literature, we had a little bit of transposon mutagenesis information, and we tried to make a hypothetical minimal genome based off all of that, and it didn't live. So we zapped our Frankenstein's monster, and lo and behold, it did not rise from the table. So we were sad. So then they basically went back to the drawing board and decided they were just going to do transposon mutagenesis. At this point...
For that section where they said it didn't work, I like how they ended with, but while we were doing that, we got really good at all these other techniques. They tried to make some lemonade out of it.
Yeah, that's a nice positive spin. We're like, but it wasn't all for naught.
But again, also that unromantic thing, at some point you do actually have to test the literature precedent for what constitutes non-essentiality. So, you know.
Yeah, absolutely, absolutely. So, you know, I like their intro to the next section where their first attempt, no good. And then their intro to the next section says, it was clear from the limited success of our hypothetical minimal genome design that we needed a better understanding of which genes are essential versus non-essential. We're like, well, that's very plainly said. And so then they moved to using TN5 mutagenesis. And so they talk a little bit about how they do that. And essentially what they're looking for is they do a lot of transposon insertions in lots of different places in this synthetic genome. And then they try to, those transformed cells, they're going to grow them. So the idea here is that if the cell is able to grow and it has a transposon inserted in a gene, you can probably conclude that that gene is non-essential because it's been disrupted and the cell still continues to be viable. So that's one of the ways that they're going to be defining essential, non-essential, quasi-essential. We'll start to get into that. They did this other thing which I thought was interesting. So they've got their initial transposon mutagenesis, where they've inserting these transposons into genes and then seeing if those clones will grow. That was what they called their P0 or their P0 pool. But what they also did was they did a few serial passages from that pool of cells to try to see if they could identify some cells that were slow growing. So basically, I think if I understand this right, they were looking for cells that kind of disappeared out of the pool after about 40 generations. Does that sound right to everybody?
I'm not familiar for this, so I was actually going to ask about that because I didn't know whether slow growing was something that they were actually like quantifying a doubling time for. So I haven't looked at any of the supplements. Is there like growth curve data that they've published with this, or no?
I have not. I have to admit, I have not looked at the supplementary information either. I don't know if they've got growth curve data in this place for the pool, this P0, P4 that they're talking about.
I would expect it for hidden versions, you know, if they were going to go that way. But I mean, I will accept that they were looking for slow growing, meaning they disappeared if it's not being done by whatever, a doubling time. That
was my impression just from the text that they've got here.
Yeah.
Is that they basically said, we're going to do serial passages, we're going to go out 40 generations, and whatever disappears from the population, we're going to conclude that those were slow-growing mutagenized cells. Anybody got any other thoughts on that?
So the ones that disappear from the population are defective for genes that are important for growth.
Yeah, I think that's a good way to put it. So the genes for the cells that disappear out of the population over the course of 40 generations, those disrupted genes are going to affect the growth rate rather than just the binary of viability.
Right.
So viability being will it grow or not, but those genes that they're talking about here are ones that are actually going to impact the growth rate instead of just viability.
Right. It's just the negative selection process of the transposon mutants can get confusing because you're selecting for the opposite phenotype of what you think it is. Yeah.
I know what you mean. I think when people first read this, you have to sit and think about, like, what are they actually going for here? So the outcome of that mutagenesis, that transposon mutagenesis, having their initial populations. Joel, did you have a thought? I saw you.
I was just gonna say something about this is where they come up with necessary versus or non-essential versus essential, and then these slow growing is where you come up with a third category.
Exactly. You beat me to it. That's exactly where I was going with this.
All right. Seemed like the next thing to talk about.
Yep. So the outcome of the P0 and the P4 in this transposon mutagenesis was that, just like Joel said, this lets them classify genes into kind of three really broad categories that they're going to call essential genes. So these are genes that they never saw cells in their population that had transposon insertions in those genes. And that is because, as Karen has pointed out, you have to, this can be counterintuitive to people. That's because if you got a transposon insertion into an essential gene, that cell is not going to grow, it's not going to divide, and therefore you're not going to find it in the P0 or the P4 population. So genes that they never saw transposon insertions into in the population of growing cells, they're going to conclude are essential because they can't be disrupted or the cell's going to die. I thought it was interesting that they make some notes about some of these genes, they saw some sparse transposon insertions in the terminal 20% of the 3' end of the gene, or the first few bases of the 5' end of the gene. So, and this, I did a little bit of stuff with this in my postdoc, which I thought was kind of interesting about the tolerance for mutations towards the end of a coding region. It seems like you can see the end of the sequence sometimes, in my case, I was looking at insertion-deletion mutations. It seems like there was a higher tolerance for those types of mutations at the end of a gene. And it sounds like they might have seen a similar thing with the transposon insertions here. But they really don't get into that too much more. And so then they're going to define as non-essential genes that they frequently see transposon insertions in both the P0 and the serial passage. So they're seeing it in their initial population. 40 generations out, they're still seeing lots of insertions into these genes. So they're going to classify those as non-essential because those genes can be disrupted and the cell still seems to be growing fine and the growth rate doesn't really seem to be that affected. And then their final category that they talk about is this quasi-essential, which is genes that they're primarily seeing them with transposon insertions in P0, and then they disappear out of the population and they're not seeing them in P4 because the implication is that they have an impact on the growth rate, but not necessarily just viability. So that's their quasi-essential. They then try to define that a little bit more into minimal growth disadvantage quasi-essential and severe growth defect quasi-essential. So they're kind of trying to define the spectrum of quasi-essential, and that got a little wobbly for me. But I don't know, do you guys have any thoughts on this idea of quasi-essential and trying to kind of nail that down?
A layer of metabolomics would make this beautiful, I guess. I don't know, I think, or confuse you more. I mean, I think if you're in the right culture medium, your NMR data or mass spec or whatever you choose, to just see, like, is something building up or not. I mean, that again, it's the biochemist thinking about how that subdivision within the quasi-essential category would be. Because, I mean, is it a protein-protein interaction that's just not happening as efficiently as you'd like because there's a bunch of other crap that you took out? Is it direct substrate utilization? I mean...
Yeah, I think at this stage, it's if I understand it right, it's a single, they're looking at clones, so it should be a single transposon insertion into a gene of the synthetic genome.
Right, but every transposon insertion creates a genomic context, right? So what's
Right.
everything doing because that thing's not there, right?
And you are starting with the synthetic genome, so yeah, that's true.
Yeah. And you also don't know there's a lot of things we don't understand about regulation and chromosomal condensation and things like that, where it might not be a protein level effect. It might be some weird, you change the shape of the chromosome, or you change something where it matters, and it changes expression of a essential gene.
Yes.
So there's a lot of stuff that could be going on where mysteries of the cell still.
Yeah.
Well, I think that right now their goal is to get the minimum genome, but once you have that, the next goal would be to use that as, in synthetic biology terms, as a chassis for putting new stuff in. And so the growth rate is quasi-essential because these wouldn't be useful organisms if they take ages to grow. So it's just I think that's why the growth rate comes into effect, is for downstream applications.
Mm-hmm. Yeah, and I thought I'd really quickly show their little. Here we go. Let's see if I. So that's the. I don't know if you guys can see that, but they have a little. Figure 3 kind of walks through what they're talking about with non-essential, essential, quasi-essential. So you could kind of see an illustration of that, with each of these little triangles indicating a transposon insertion. And so what they're looking at here is they're kind of showing the different categories that they found in this SYN1, so their synthetic genome. So they're finding the majority of the genes they're going to classify as non-essential. Then they've got about 240 that they're classifying as essential because they don't get any insertions. And then they've got this quasi-essential space that they're going to try and define based on growth rate and so forth. So I thought that was a reasonably good illustration of these, at least what they're seeing from the Transposon Mutagenesis library. So they started out here, and I think it's interesting that they towards the end of that section they talk about they did something that they called a design, build, test cycle, which parallels what they did with the synthetic genome in their 2010 paper. But towards the end of that section they say, well, we also took the traditional sequential deletion approach to genome reduction. We did stepwise deletions, so forth and so on, but yeah, we don't want to do that. So we're going to move forward with the design, build, test, which to me, after you read through all the remaining sections, sounds kind of painful. Like, I'm really like, it's like Mary Shelley wrote this. Like, let me just throw my stuff in here, and let me just zap it with some electricity, and it'll stagger off the table and start to learn French or whatever, But
everything's on fire in the end.
Yeah, and everything's on fire in the end. Exactly. That's how it ends. But they basically went through a lot of rounds of trying to piece this thing together. And I think Nicole might have said it's like copy and pasting in Photoshop. So I don't know if I want to kind of launch into, if anybody has specific things they want to ask about in those sections, feel free. But I think we could kind of really break our brains trying to kind of piece together exactly what they did. What I do think might be useful is to look at, let's see, I'll put this up. This is the, there we go. There we go. Let's do that. This is a table at the end, which kind of just talks through the different stages that they did this in. So what we've got here, I don't know if you guys can see the arrow as I move it around, but what you've got here is they're starting genome, the synthetic genome, the SYN1, and it shows you the genome size and it shows you some information on the growth rate. And then they kind of talk through the different steps that they did to get to their final product, which is this one down here, SYN3, which has these 8 segments that they have kind of tinkered with and copied and pasted and did all this Photoshop work with. And the outcome of it is a 531 kilobase pair genome with 473 genes. So we're going down by a little over half from the original. So here's the original with 901. The final minimal genome that they've put together is 473. And you can see you've gone down by about half in the size too. Doubling time's a little bit longer, so it's 60 minutes for the original SYN1, and now we're to 180 minutes of doubling time for this final product. There is a lot, as you can see, that went on in between those. And they do discuss this a little bit, where they talk about what they pulled out, what they pieced together, what they kind of tried to do. And I think the main thing that jumped out to me with this that I thought was important was the idea of redundant genes for essential functions. So there's a whole section that I think that concept is worth pulling out from all this technical detail about how they did this, which is the idea that they are very aware that you have, in a naturally occurring bacterial genome, you're going to have a couple of genes that might fulfill the same function. And
can we qualify, sorry.
No, please.
The redundancy, so I'm clear on it, because I did read the last half of this paper in a rush, is the assumption that 2 genes with the same functional annotation were considered. I guess I want this criteria for redundancy. Was it based on a description of what the thing was doing, or sequence similarity, or something else?
I don't think they specified.
Okay.
If anybody else read them define what they're going to call redundancy.
Yeah, because they say they may or may not be paralogs. So, but,
Joel?
I was just going to say, if you knock both out, it stops growing, and that means, but if you have one or the other, it grows. So that's pretty much the end of their definition of redundancy.
Okay.
Especially where they don't know the function of a lot of these genes in the end.
Mm-hmm.
If it's a phenotypic thing, it doesn't need to be.
Okay.
I mean, you can think of POL1 and RNase H, right? Like you can knock POL1 out, and RNase H can kind of take away primers and replication. So there's no similarity there.
Okay.
There's no functional similarity there, except that they phenotypically kind of do the same thing.
Yeah, I guess I'm kind of overthinking this a bit, so I apologize. It's just I was wondering.
No, I think that's a really good question. Because, and I think it gets to one of the reasons why I think this is really interesting, the idea of the redundancy, is because, I mean, to me, that's an entire angle to take on what they're doing with these genomes, is pretty much, Nicole, kind of I think maybe what you're asking, which is, you've got redundancy in a genome. What is the nature of the redundancy? You know, how many times do you have a redundant gene pair because they're paralogs? How many times do you have convergent evolution, and therefore now you have functional redundancy? What's the selective pressure that leads to functional redundancy? Why would you still retain 2 of these? What, you know, I mean, you could just like start tripping over yourself asking questions about why does Mycoplasma, of all things, still have stuff that's redundant? Yeah, so this basically was one of the things that they had to deal with, because their method of doing this in 8 segments, so they split the genome into 8 segments and then kind of tinkered with each segment independently. They did potentially run the risk, and they talk about this happening, of deeming a gene non-essential on one segment and another gene non-essential on another segment when they are in fact redundant. So if you take them both out because you think they're non-essential, now suddenly you've lost viability because you didn't realize that those 2 genes, one of those needed to be there. So they talk a little bit about trying to deal with that. And I don't think they quantified it though, did they? Did I miss them saying like we deemed this many to be redundant, or did they just kind of like, I think they just talked about it as a concept. But yeah, I just think that in itself I think is really fascinating. You could, I mean, there's all kinds of molecular evolution questions you can ask there too, about selective pressure and rates of evolution and so forth, which I think is cool. Okay.
So what I'm looking forward to with this, the study that somebody needs to do. So if you just took these and passaged them Lenski style for a while, like how much better would the cells get at growing, right? Like so they're taking all these design principles and trying to get it to what's the best doubling time, but just delete things and let the cells tell you what's wrong. Like,
do you think
I don't know
they're
if
doing
they're
that?
gonna. I don't know. It doesn't seem like what they would do, but I don't know. It's a right down the middle enough pitch where somebody should do it, right?
Yeah. Yeah, I agree. I agree. Okay, so they end up, as we mentioned, what they end up with is their SYN3, which is the minimal genome. That's what they're talking about in the title of the paper as being the minimal genome, is the SYN3. And then they spend a bit of time, and stop me if somebody's got a methodological question that you really want answered. Otherwise, I'm going to kind of launch into like what they found in terms of the types of genes and so forth. So then they kind of talked a little bit about, well, okay, so what's retained and what's not retained, and what have we got in terms of categories of genes and so forth. So they talk about, there's a section where they kind of talk about the descriptions of the genes. And let me flip up to the figure. So they split the genes that are present in the SYN3 genome. So in their minimal genome, they've split all the genes up into 5 categories, which are called equivalog, probable, putative, generic, and unknown. And the equivalog, I think, is a terminology that they use back in Tiger times when they were running their annotation pipeline and so forth. So equivalog is basically, if I understand this figure correctly, I think these are each column, there should be like little columns here, I think, and I think each column is a gene in the SYN3. And so you've got, 232 genes that are equivalogs, 58 genes listed across here that are probable, 34 genes that are putative, 84 that are generic, and 65 that are unknown. So what they're doing now is they just ran BLAST-P, and they used a pretty a low E-value here. And they basically said, do you see this by sequence similarity only? That's all we're looking at. Do you see something similar in all these other organisms? So they've got Homo sapiens on there, they've got Drosophila, and then they've got E. coli, Staph aureus, and then some Mycoplasma non-mycoides. And so equivalog-wise, I mean, you got a lot of similarity generally speaking, but where you have white space is that there's a gap where there's no obvious similar sequence in these other organisms. And obviously what's going to happen is that white space gets kind of bigger as you start to get stuff that's not defined as well. So generic and unknown are these 2 categories where they're kind of like, we don't really know. That's generic. It's probably a kinase of some kind. And then unknown, they're just basically like, I don't have any idea. And this is the category which causes me to have palpitations because I just don't, what do they do? Why are they there? What's happening? Nobody knows. And we don't have a lot of information from other organisms. So that's a big open question from this, I think. And it's like, I think, they do talk a little bit about sequence divergence. So I don't actually know that much about rates of evolution in Mycoplasma. Is there an expectation that sequence divergence would be high enough that you could see a homologous sequence just kind of off on its own long branch that you're not going to recognize it anymore?
No, I mean there's enough annotated within like genitalium and other Mycoplasma and decently closely related things where you're not gonna. I mean, all the Mycoplasmas are weird, but there's enough out there where you should see something similar. Yeah, I mean I think the take-home message that a lot of people took from this is that we don't know a lot about a lot of the genes, even though they're essential, right? kind of bringing together this Michigan meeting too. So Trina McMahon gave a great talk where the part of it was going into looking at metabolic models and gene models and predicting species interactions from annotations and genome annotations. And I just couldn't help worrying about, like, we're not good at annotating things, and we're not going to be good for a long time. Like, there's, you struggle with questions about what's the best way to annotate a genome. Well, the answer is that they're all crappy. Like, there's no best way. There's different poor ways to do it. So I don't know. And I talked to, thinking of like building metabolic models for things and building these kind of flux-based models. It's amazing that they work when they do, and it worries me, like, if I ever want to jump into it, like, I just don't know how much hay I need to sort through to actually get to the needle there. And, it might be a bunch of needles, it might be one needle that's covered by a bunch of other stuff. So it scares me in terms of, like, getting into these larger systems biology questions, because even in this minimal genome, like, we really, and with all of our knowledge from 40, 50 years of microbial genetics, like, we can't predict what the heck most of these things are doing.
Yeah, I mean, I agree with that. I think that giant white space is like, I mean, that is a giant black box. You still now, even like you said, with this minimal genome, you've broken it down to what you're going to deem to be at least quasi-essential, if not essential. And you still got this black box that you're just like knocking at, going, what do you do? We don't know. We don't have any idea. We don't even know if you're a kinase. We've got nothing. We have nothing. And that is pretty surprising. I mean, and to your point about metabolic models, that even for stuff we do know, there's always the messy. I always worry about kind of like transitive annotations, So this is a kinase, sure, but it might not be that kind of kinase just because A looks like B, looks like C, looks like D, so D must be A. You're like, oh god. So that gets messy too. Yeah, so I agree. I think it has a lot to say about what we can conclude from trying to do metabolic modeling and so forth. So the other thing that they do is they talked a little bit about functional categories and whether they were kept or deleted in their final minimal genome, which is table 1. So I'll just pop table 1 up there really quick because you guys can see that? Yeah. Because what they've got here is the list of functional categories down there, a big list, and whether or not the gene in that functional category, how many genes were retained, how many genes were deleted. So this is going to be our final gene count, 473 for SYN3, the minimal genome. If you add kept and deleted together, that's the gene count for SYN1, which is where we started. And what you can see is some of these things that were completely removed are very predictable. So mobile genetic elements and DNA restriction, they just jettisoned that. Here, transport and catabolism of non-glucose carbon sources. They talk about this in the discussion where they're basically, and Nicole mentioned this, like, you're just going to feed this thing glucose. So it's going to keep all of its glucose stuff up here at the top, and then it's going to pretty much get rid of a bunch of stuff that has to do with dealing with unusual or non-glucose carbon sources. This is a very stable environment that you're growing it in.
So I don't have the paper in front of me. What's the media they're growing it in?
Oh boy, that's actually a really good question. I don't know.
They do say rich growth medium, which is not what the answer you want.
Yeah, I mean, so that's the thing, right? If it's too rich, like, you're gonna, it's almost easier for these minimal guys to grow them in poor growth media because they don't get swamped with nutrients and kind of go crazy with it.
Oh yeah, that's interesting. I hadn't thought about that. That's, yeah. I actually didn't look at that. I don't know what they're growing it in, to be honest. I actually don't know what Mycoplasma is happy in regardless. I have no idea.
It's happy in goat.
In what?
Goat cells.
Goat cells. Oh, all right. Well, there you go. That might be why I've never grown it. I don't really have a purveyor of goat cells. I think the upshot of this is that it keeps a lot of what you would expect, which are kind of things that have to do, and they mention this, they kind of break this down a little bit into, I don't know if this makes anybody dizzy, sorry. In figure 6, they kind of break these categories down into 4 sections So they're like, you know, expression of genome information, that seems pretty important. Preservation of genome information, that seems pretty important. So information processes, those genes were by and large retained. They talk a little bit about cell membrane, so it's going to keep some cell membrane genes. Mycoplasma is cool because it doesn't have a cell wall, so they don't have to worry about that. And then they've got cytosolic metabolism as being another category, and then unassigned, which they kind of say, once we figure out what these are doing, they're probably going to fall into one of these 4 categories. So, I don't know, a little blue piece of the pie, you'll eventually come into these. And so that's kind of what they break the table into, is to looking at how these specific functional categories break into those 4 broader sections, which kind of makes sense if you think about them. Those are the processes that you would think would be categorized as essential.
The thing that I thought of was, I was looking through that list, is that viruses, the main point of a virus genome is to basically get to mRNA, and then they just use the host's translation machinery. And for translation, there were 91 translation genes, and only 2 of them were able to be deleted. And so that would be 90 genes every virus would need to carry around. So that's why they just like to hijack the host's translation. That was my thought.
Yeah, true. Ooh, I think I'm getting an echo off of somebody. All right. Okay, so there we go. Thanks. Yeah, Jill, you're right. Yeah, so yeah, they've got this. There's their translation category right there. So 89 genes retained, only 2 genes deleted. So yeah, I think you can think a little bit about essential processes and the idea of viruses. That's true. Yeah, so that's kind of where they go with this is, and then they've talked a little bit in the last couple sections of the paper. They talk a little bit about kind of some morphological changes that they observe in the cells. And so I thought that was interesting, because not only do we get a difference in doubling time, but then they're also seeing some kind of what look like smaller cells that are apparently viable, that they'll go through a 0.2 micron filter. And I have to say I wasn't entirely surprised by this, because we are running across this in my lab with marine Vibrio that do this. It's very annoying, because we're trying to feed them to our marine predatory bacteria, and we need to be able to filter them out sometimes. But apparently marine Vibrio make something called little mini cells that make it through our filters and then end up polluting our filtrate. We're very annoyed about that. So I wasn't entirely surprised. There's my little poor man screenshot that you get these little mini cells. But that's an interesting question that I think they're going to follow up on too, which is, why do you see these morphological changes? What does that mean in terms of the genes that they've taken out? Where might this be coming from? And the vesicles were interesting too. I don't know anything about that. They're seeing more filamentous cells, more vesicles. Don't know. So I'll bet they're hopefully looking into that.
I'm going to throw out a very poorly informed hypothesis about that, but it's based on some other things they said about, removing cytoskeletal elements. And I mean, cells are going to do other things other than, these strategic changes in the evolutionary landscape of their genes to accomplish the business of staying alive. So maybe it's an alteration in cell morphology and topology to facilitate the macromolecular interactions that need to happen for physiology, because contact times, on-off rates within that cytoplasm aren't happening in any other configuration. You've removed membrane components, so I don't know what that does with respect to topology and temperature response and distribution of transmembrane elements. So changes in shape to kind of work with what you've got. And I mean, this is a question I've actually been asking myself in an area that's very different from this, but I had a discussion with one of my supervisors last week about arbuscular mycorrhizae, which are fungi that kind of they grow these very convoluted internal surface areas as they penetrate into tree roots. And the question is, why do you spend such a metabolic load on expansive cellular interface. And my question is, well, what if this is the trade-off because they have perpetually crappy transporters with bad rates? Just give it enough carpet so that it can still get what it needs if the kinetics are bad. But I don't know if the kinetics are bad. That's just my idea, because I don't know what protein repertoire feels like. Although on the lifestyle does seem like a whole lot of waiting around for stuff to come in. It's a lot of diffusional type things, and using your secretome to get things back into you so that you can provision to your host. So those are my thoughts on shape.
Yeah, I think that's an interesting idea. Certainly, as you said, there's going to have to be trade-offs, and this might be one of the things. If you're only going to look at the genome and you're only going to see do you get cells or not, you might miss important things like this, where you're actually seeing phenotype. You can measure some phenotypic differences and some phenotypic changes. Yeah, that's cool. I hope they're looking into that.
Mm-hmm.
I have something to add in terms of shape, which is I heard a talk by another person at the University of Oregon Meta Center, their System Biology Center, which Tristan Ursel, who works on he worked on bacterial shape and MERV, and how MERV can maintain pressure of the bacteria and helps maintain its shape. So in terms of the mycoplasma, because there must be some sort of pressure that needs to be maintained within the bacteria, and if those genes are deleted, then there are probably other physical forces that are involved that are preventing the bacteria from being like maintaining its hydrostatic shape. So it could be a reason why you see them as smaller rather than as bigger, because they're missing those key components in terms of being able to maintain the pressure, and so it's probably more beneficial or thermodynamically favorable for them to be smaller.
I muted myself, and then I was like, "That's cool," and then you couldn't hear me say that, so I'll just repeat myself. That was cool. Yeah, that's a neat idea. Just there's something that I don't think about very often, which is like the physics of being a cell. I think that's something you have to think about in this context. That's a really good point. Okay, well, I'm always reluctant to keep people more than an hour because everybody's busy. They talk a little bit about a couple other things. Can I get Nicole to maybe mute herself? Sorry. Okay, cool. I hear the echo and then I get distracted. They talk about a couple other things that they're doing, which I think are more like previews for things that they're thinking about, like changing the ribosomal RNA gene to try to figure out what effect that has. Doing a couple of different things that sound kind of neat, but I don't think are fully developed yet. They're following up on codon usage, for instance. Mycoplasma is neat in that it uses TGA as a codon for tryptophan instead of being a stop codon. And so they're going to potentially do some things with that now that they have this minimal cell to see what happens. And then I think that's where they're going with this. We talked a little bit in the beginning about overall where they might want to go with this, which is, what are the functions of some of these genes that are unassigned right now? And then also, can this be the scaffolding for trying to build something up that in a very controlled environment could be an efficient way to produce things in an industrial process? Could this be something that you could use as a framework? And I think that's kind of where they're going with some of this. So I thought I would ask, kind of, the discussion goes through a couple of these things, but I thought I would ask to kind of close out if anybody's got any thoughts on, where do you think this is going? What do you think is the most likely thing we're going to see next from SYN3? Now that they've got it, it's in their freezer, they can do whatever they want with it. Where do you think is the likeliest place that this is going to go?
They're going to start putting other stuff in it. That's my guess.
Yep, I think that seems likely. Joel?
I was just going to say that this is away from SYN3, but it was in the news today or for the past week. There was some big closed meeting of people talking about synthesizing a human genome. And I don't know if they'd be using these sorts of techniques that they've kind of perfected in this paper or not, but it was George Church and some other people, and they had it a closed meeting because it was supposed to be, they were also discussing a paper that hadn't been published yet, but they're mixing both talking about that paper and talking about generating the human genome in the same meeting. And so there was some backlash as far as why would you have a closed meeting to talk about generating an entire human genome with the idea that you could make a new human without having to go through the typical reproductive things. But anyway, I just thought it was kind of timely with this discussion of taking a synthetic genome and putting it into a cell and having it continue and live on. But I don't know where else to go with that. But.
Also gonna, so if you look at the scaling up, anything with the centromere is going to be really hard to do this way. It just we basically don't have a complete human genome sequence yet, right? Just because maybe that might be now, but we basically haven't sequenced through the centromeres of a lot of things yet because it's hard. And so synthesizing those regions is no easier than sequencing them, basically. And so it's easy with bacteria. I'm not sure how it scales up to other things, but
yeah, and I think the very first part of the discussion kind of struck me, where I think it might be at this point at least, perhaps ambitious, where they say genomics is moving from a descriptive phase to a synthetic phase in which whole genomes can be built by chemical synthesis. It's like, perhaps not yet. I mean, I don't know that this is going to become commonplace anytime soon, but one can hope, I suppose, if you want to be using this as an approach to try to get at some of these questions. Karen and Nicole, did you guys have any final thoughts you wanted to share?
Keeping in mind context, I want to see them stick in a bunch of reporters or tags, and I want to watch movies. I want to see the interactum. I want them to do some visuals. That would be kind of fun.
Yeah.
I'm also thinking about orchestrating pleiotropic responses when you cut things down that much. I don't know how you would look at that, but that's the other thing, right? I mean,
can you actually request this from them, or is this? Can you send me your Sin 3, please? You know, they'll be like, no.
I'm gonna throw out a random guess and say it's patented. Just gonna throw that out there.
It what?
I think it's patented.
Patented it. Yeah. Okay. That would make sense. Yeah. Well, there you are. So on that note, nothing's on fire. So our version of Frankenstein has ended in a much more positive way. So thanks everybody for being here. I thought this was. I'm really glad to. I think Joel suggested this. And then I actually got an email from somebody who said, you know, would you guys maybe do this paper? So 2 votes for the same paper. We had to do it, and I'm glad we did because I hadn't had a chance to read it, and it was good to kind of sit down with this and really think about what they were doing and where they were going with it. So thanks everybody for being here. I know it was kind of short notice, so I'm really glad that we got a good turnout. At this point, what we're going to do with the Journal Club in the summer transitions into a book club. I think I've mentioned this a couple times before. Last summer we did the Summer of Symbiosis. This summer we're doing the Summer of Microbiology Books for the Public. Always self-serving so that I can pick a new book for my non-majors course. The first book we're going to do in June is Pandemic, suggested by Karen, and I will very, very shortly get that information posted to the website, so if people are interested, you can get the book ordered and get a chance to read it. We'll probably do it in mid to late June to give people a chance to read the book and so forth. But I hope that people can join us for that. Joel.
I've been listening to the audiobook.
How is it?
It's a little bit strange because it's read by the author. She has a very soothing, nice voice, and she's talking about these super scary topics. And there's also, for whatever reason, there's just a very slight reverberation or something like that with the audio, and so there's like a tiny bit of an echo, and it bugs me like crazy to have that little bit of echo in there. But I still enjoy having the soothing lady tell me about pandemics.
Yeah, when you started saying she was very soothing, I just started getting in my mind something like, and then everyone will bleed from the mouth and die. And you're like, okay. So maybe that's there's a preview of the audiobook review if you're thinking about getting the audiobook for that. All right, so that will be in June. I will get the information posted shortly. Thank you to everybody for being here. Hooray for everybody who is now into their summer and not in the academic year. And hopefully we'll see you guys in June. So, thanks.
Thank you.